Thanks, Romina. Thank you for the invitation to speak here, and thanks for pitching up this morning after such a wonderful evening last night. I'll just wait for the first slide to come up. Great. Okay, so I've got a lot to cover in 15 minutes, so just bear with me. Right, so firstly, what are we talking about when we talk about ATMPs, or Advanced Therapy Medicinal Products? Well, as defined by the European Medicines Agency, they're simply gene therapies or ASO therapies, and largely speaking, somatic or tissue-engineered medicines, which are largely cell therapies. Now, we've heard over the past few days the vast amount of money that these trials cost. What we haven't heard about is the vast amount of money it takes to develop these ATMPs before they ever get to clinical trials. But there has obviously been a huge amount of investment in this area. There's a huge amount of help and regulation by the EMA and the FDA. And this has resulted in a huge amount of these trials coming to clinic, which is wonderful. But the point I wanted to make with this slide, after I walked around Bologna the other day and took a little photograph, is that poor old Atlas there is Mr. Delivery. And we're depending a lot on delivery to I suppose, shoulder the burden of all these trials. And there has not been the same amount of investment in research into what I call delivery science. So why direct delivery? Why just not give it into the bloodstream? Well, as Romina has said, and many people know, the brain has a structure called the blood-brain barrier, which prevents a lot of substances entering the brain to preserve brain function. So we just simply can't, for lots of therapeutics, they simply won't get into the brain in the concentrations we want. We also, of course, will want to deliver to focal neuronal targets or neural targets, for example, the striatum. But we may also want to deliver to targets more widely. And we can do that because of the connectivity of some of these focal targets. So we can actually target networks as well as focal areas. And the last point to make here is that one must be careful of off-cell on-target effects. So the brain replicates a lot of its signaling mechanisms through different parts of the brain with the same molecules at different concentrations and doing different functions. So if you knock down or knock out something in one area, it may have unwanted effects in the rest of the brain. Again, an argument for doing focal delivery. So what are the challenges to targeting? Well, accuracy is the first one. This has been really overcome. This is Lars Lexell and his stereotactic frame, which co-register the patient's head, the patient's space, the operative space, with the imaging that guided the surgery. These days it's a little more slick. We don't have this great big piece of metal in the way, and so we use these infrared-guided, image-guided systems. So our instruments in the patient's head are registered, and the preoperative imaging is all included. And we now, of course, have nice robotic systems which help us as well. So what about the next challenge to targeting? Getting there in pathological brain. Here's a normal brain, okay? And we want to target the caudate and the putamen. So we've got a nice target, a nice pathway here. We're running in. We're not crossing any sulci where all the vessels are. We have a nice long run in into the head of the caudate, and that would make for a very happy neurosurgeon. Similarly, we can target in this direction, getting into the putamen, entering at the apex, and then we have a nice long run to deliver our therapy into the target. This is an MRI of a patient with HD, in which this task is a little more difficult. Here, you'll see that because the ventricles have dilated, we're moving our trajectory out quite far to get the similar run into the caudate head. And when we try and get into the putamen on this side, we're invariably going to come across sulci that have been dilated. So it's not as easy as it seems, and ventricular dilatation, atrophy, and sulcal limitations are a real headache for us, and requires a lot of planning. The other headache we have, of course, are blood vessels. Well, blood vessels are always a headache, but when your target atrophies, your blood vessels don't, and therefore the density of your vessels increases and makes you more likely to hit them. The next problem is what I call getting there but going elsewhere. So in this MRI scan, you can see that we've got perivascular spaces at the bottom of the striatum. These are present throughout the brain. They're often potential spaces. And the problem is when you infuse, as we can see here, then some of the infusate will suddenly get into a perivascular space and just simply disappear. And this is really relevant in Huntington's disease, because there's a greater amount of perivascular space dilatation in advanced Huntington's disease. So it's a particular problem for us. What about how to get the ATMPs in the first place? Well, way back in the day when I had hair and was a lot slimmer than I am now, people just tried to put in cannulas in the brain and deliver by direct flow. And the volume of distribution was never greater than the volume of infusion. And basically, it didn't work. What we now know is that people like Paul Larson and Chris Bankovich developed convection-enhanced delivery, where if you have a structure on the catheter which prevents or reduces reflux, and you deliver the therapy very slowly, you can then expand the extracellular space, and the volume of diffusion is much greater than the volume of infusion, and that makes this thing technically possible. A process called convection-enhanced delivery, but it takes time. Okay, you have to do very low infusion rates. So these are the kind of technologies we have for modifying the catheter designs. This is the catheter design manufactured and sold by ClearPoint. And we've got a two-step design here with a step here and a step here. And what happens is your therapeutic comes out at the end, hits the first step, and expands. Then it gets beyond that step, hits the second step, and then slowly over time expands again. Eventually, though, it will start to reflux. So with the ClearPoint system, what we do is we advance the catheter and we repeat the whole process, again coming out at the distal step, then going back to the proximal step, and then expanding into the perivascular space. The Renishaw system uses a slightly different technology where it has a recessed seal here in the catheter system, so tissue gets stuck in here. And in this system, we get your convection-enhanced, your therapeutic just comes right out, stops at the recessed seal, and then because that seal is very competent, then delivers the therapeutic again in a CED device, in a CED-directed manner. The difference here is the catheter system is fixed, and you don't have to advance it. My disclosure is, which I completely forgot, is I consult for both ClearPoint and Renishaw, and I'm doing the Unicure trial. So I'm agnostic. I don't really care. I don't have a preference. All I care about is what works. So how do we know when enough is enough? Well, we use intraoperative real-time contrast monitoring. And here's the kind of technology we use. This is the ClearPoint system. Looks like something you get in the middle aisle of Aldi or Lidl, but it works. It's really cool. Here we have it onto the patient's head under anesthesia within the MRI scan. And here we are drilling some of the holes to get the catheters into the patient's head to deliver the therapeutic. So for this fluid-based therapy, for convection-enhanced delivery, we have here our initial bolus in the head of the caudate here, and then over time it increases, and all looks great. Okay, so it's reassuring. However, how useful is it? Can I just drag you all back to high school math for a second and remind you that the volume of a sphere is proportional to the cube of the radius. And this curve here is the relationship between the radius and the volume of that sphere. These curves are if you increase the radius by 1 millimeter and by 2 millimeters. So look at a sphere with a radius of 3 millimeters. Normally it'll be about 100 microns will be the volume. You're going to have to add, for that to increase by 1 millimeter, you're going to have to add another 100 microns of volume. However, to get it to go to 2 millimeters larger, you're going to have to add 400 additional microliters of volume. So therefore, the radius or the diameter of the sphere isn't particularly sensitive to the amount of volume you're actually delivering. Also, the problem with gadolinium is its brightness isn't proportional to its concentration. Think of it as an overexposed photograph. So again, it's hard to really work out how much of the therapeutic you've actually gotten in. Lastly, but certainly not least, you're not delivering a sphere into a vacuum, into space. You've got a tissue structure, and you're delivering it into the extracellular space, which ain't spherical. However, this is the best we have at the moment, but we could do with a bit more modelling and a bit of greater understanding in this area. What about cell therapies? Well, this is to remind people, John Steinbeck's of Mice and Men, that a lot of the technology for delivering cells has basically been just simply upgraded or amplified from small animals, and quite frankly, it doesn't really work very well. This Nice Review article was one of the first to really show that when you try and put cells through a syringe, then you get significant shear wall stress, you get significant deformation of the cells as they enter the actual needle, and you get acceleration and deacceleration injuries, all of which can affect cell viability, can affect cell differentiation, etc. So really not what you want to do to your cellular therapeutic, right? So we've developed some systems, and this is just some unpublished work I'm just sharing with you, where we made gels that have the same consistency as brain, and we use bioluminescence, where the cells emit light in proportion to their ATP production, so we know that they're alive, and this is a real-time readout. And then we can look at different methods of delivering cells, either by delivering cells and then withdrawing, delivering more cells, called the withdrawal method, or we can try and inject cells into a preformed track. And by nature of the level of the bioluminescence, we can measure the amount of reflux, the number of live cells, and the cell density that we're delivering in real time. This is a busy slide, but it's just to show you using the withdrawal method where the catheter moves and the preformed track method where the catheter remains the same. These are the boluses of live cells that we're delivering into these gels. And you can see here that with the withdrawal method, then different types of catheters perform differently. So one with a short step creates more reflux than one with a tissue seal or a longer step. Similarly, with the delivering into the tract, only the catheter with the seal actually minimizes reflux. And if you look at the cell density of what we're delivering, the preformed tract level method gives you less cells per deposit than the withdrawal method, except for the recessed tissue catheter, which appears to perform equally well in both. Okay, and we took this into a porcine model and again found exactly the same thing, where this is the amount of cells that were delivered to the striatum with a recessed catheter track, and this is the amount we got with a stepped catheter with most of the cells having reflux back up to the surface. So what about optimizing distribution? Another problem pointed out initially by Steve Dunnett, we've done some more work on it, is about how the cells sediment. So if you have a cannula that's horizontal, all the cells will tend to come out at the end of your delivery. If you have a catheter system where the cannula is vertical, they all tend to come out at the start. Again, this is a bit of a problem. Okay. Draw your attention to a lovely bit of work done by Dan Lim's group in UCSF, where in order to get the distribution, the cells more widely distributed within a structure, they have a single penetration and use a radial catheter that they can just stick out into the parenchyma of the brain and rotate and withdraw and rotate and deliver the cells up in a kind of a three-dimensional spiral into a larger structure. Certainly clever stuff, but I don't think it's been used in anger in human brain yet. Okay? What we really need is to know, to be able to clinically track these cells. And these are a number of recent review articles which are trying to bring some of this technology into the clinic, either with MRI and APSOs or with PET and zirconium labeling of these cells, so we can track them in real time and make sure they're alive and where they're going within our patients in our clinical trials. Last point, and nearly finishing, talking about where is optimal. So for a gene therapy, you may want to fill the whole structure, but for cellular therapy, it may be really important to know where exactly to put the cells. They may be, the striatum does lose neurons in a dorsoventral pattern. So should we put the cells into the areas that have lost cells? Should we be putting cells into the area that still has some cells to try and support the ones we're putting in? These are questions that we simply don't know the answer to. High resolution imaging, this is a 7 Tesla scan of a human brain, may give us some answers, but again, an area that we need to be looking at. Okay. Lastly, we really need to know this important question. We all want to know this: has the ATMP worked? Okay. So what do we use? We use clinical endpoints, but they're chronic, and they're variable because of the variable disease progression. We've heard a lot about biomarkers, both imaging and CSF and serum biomarkers. Are they predictive of function? Functional biomarkers, are they predictive of quality of life? A really important question. However, a just as important question is this one. How do we know when the delivery fails early? How do we know that the delivery has worked, but the ATMP hasn't? This is really, really critical. In other words, I've just showed you a Romulan warbird uncloaking. for the Trekkie fans in the audience, probably just my age group and nobody else. But really, it makes the message, how do we make the delivery visible? Because at the moment, it's invisible. This is a key challenge in the whole sphere. Lastly, how do we make delivery deliverable, right? Our experience over the last few years setting up these type of trials and a centre for doing these trials over the last 7 years takes 2 to 3 years to set up one of these interventional delivery trials from scratch. You've got a lot of regulatory hurdles, tension with your healthcare delivery system, ethical permissions, your local GSMO committees. You need to have a really good working relationship with your CROs. This is not trivial stuff. There's a learning curve in doing this work. There's a lot of people in team management. These are long cases. The Unicure trial, the average duration of the surgery is 14 hours and can go a few hours longer than that. How do we ensure delivery fidelity, and how do we scale up beyond proof of principle once, as was said yesterday, we get the first whiff of success in this area? So this is where the whole field started. This is the Wright Flyer. I think we got beyond that. I'm trying to think of an analogy to show us where we are. So I think we're gone beyond that. We've got CED working pretty well. This is a de Havilland Dove photographed in the 1946 flying over Norfolk. This was the first passenger airline design. It worked. It wasn't easy to fly. Most of them crashed. But at least it worked. This is where we want to get to. We want to get a combination of EasyJet and Deliveroo, so we can deliver these cells into people easily, quickly, safely. As Romina said, we have the Surgical Delivery Task Force. This is our work plan. So we have our core group of people. We're cooperating with industry and with pharma and with device manufacturers, looking at procedures and these devices. And we aim to produce reports, publications, and engender collaboration. Because as was said yesterday, as Sarah said yesterday, this is a global problem. We need to work together. So these are some of the publications that we've published already. And we're going to set up a face-to-face meeting in 2023 to which everybody is invited. Okay? Thank you for your attention.